Yeah, I mean, there's a lot of really interesting things in this paper. I bet the conspiracy theorists kind of went to town with this, but for me, it's some of the particularities of the molecular details that I found most interesting, and you really get into the ACE2 receptor morphology, and you talk about how SARS-CoV-2 both appears to be optimized for binding to this human receptor, but that it's also not as optimal as the previous version of SARS. So I just would love to invite you to say a little bit about both the fit, but also the imperfect fit, and what that tells us. And I think as background, what is ACE2? Like, what else is it involved in? How much do we know about this particular receptor?
Yeah, I mean, I would say that, the background for the study initially was that we wanted to understand, first of all, what makes this such an effective human pathogen, because zoonotic, diseases don't do this typically, right? I mean, this is the first time that a non-flu virus has done this, and flu has been a long-term known human pathogen, right? So to see a new non-flu virus all of a sudden, spilling over and then just immediately taking off like this is highly unusual. So that's sort of how we got into the study, right? And saying, like, that's really weird. Can we actually explain that? And then we looked, at the genome and said that, okay, what is special about this particular virus? And the two things is this, optimal binding to the human version of the ACE2 receptor, and then the introduction of this furin cleavage site, which makes it really well at not just, so the first part, the optimal binding, allows it to stick to cells really well. The furin cleavage site allows it to then, once it's internalized into the cell, actually fuse into the cell and lead to actual infection of that individual cell. And that's probably what sort of helps explain SARS-CoV-2, because if you look at the flu viruses, they're either, really high up in the respiratory tracts, high up in the airways, where they spread pretty easily, but they don't really cause that severe disease. And then you have the bird flu viruses. They use a slightly different sialic acid, right? But they are much lower down into the lungs and cause much more severe disease. But then they don't spread very well, right? So for flu, it's one or the other. SARS was also, low down, right, but not very high up, so that also didn't transmit very well, and that was why we could get that under control. What's really unique about this guy is that it can do both, right? So this is totally new. We've never seen this before, and we think these two things explain that. So that was the first interest in the paper, right? Saying, like, why is it that this is actually such an effective human pathogen? And I should say that when we got into this, also we described most in the study, was, hypotheses, right? Nobody had actually proved that furin cleavage site, for example, is active. Nobody had proved that it really did bind better. This was based on simulations, right? But we now all know this to be true. So
could you just slow down with that just a little bit? Like, I tried to do my best, like, Google research to understand those three words: furin, cleavage, site. So site, I can understand, it's part of this molecule, but what does cleavage mean, and what is furin?
Yeah, so when these viruses infect cells, two things need to happen. The first thing is it needs to attach to its receptor, right? So the receptor is on the outside of the cell. And for SARS-CoV-2 as well, for SARS, this is ACE2, which is a host enzyme that sits on a lot of different cells, in the airways, in the lungs, in the gut, kidney, heart, you name it, everywhere. So that's the first step. The next step is that it needs to fuse into the cell. So binding to that receptor gets internalized with endocytosis, and then it needs to fuse into the cytoplasm. Otherwise, it can't actually infect the cell. Now, all of this is mediated by what's called the spike protein. It's the protein that sits on the outside of the virus, right? But the thing is, this is sort of a two-part type protein. It has the S1 and the S2, and the S1 is the one that mediates the binding. That contains the receptor binding domain, the RBD, and that's the one that's highly optimized for binding to the human version of ACE2. And then S2, which mediates fusion into the cell. Now, this S2 can only function if it's cleaved from S1. So once it's initially made, it's one single protein, right? And it cleaves in the middle, and now the virus can actually fuse into the cell. For SARS,
does cleave mean the same thing in plain English as it does in biology?
it just means like cut in the middle.
Okay.
Is the host cell doing the cleavage, or how does...
So here's why this all differs, right? So if you're SARS, so SARS-1, right? This needs to be mediated. SARS-1 is pretty crappy at doing this. Okay, so and that needs to be mediated. I've forgotten the specific enzymes now, but this occurs in the Golgi or something. Right? It doesn't really work very well, right? But it will do it. But that means that it's quite restricted in the number of cells it can actually fuse into. It can bind to all the same cells as SARS-CoV-2 can because the receptor is the same, right? But then SARS-1 sort of gets internalized, but then it gets degraded, right, in the endosome because it can't actually fuse into the cell and start the next cycle. Now, for SARS-CoV-2, when new viral particles are produced, this spike protein is actually pre-cleaved. Okay, so new virus is saying like, so that when that gets into a new cell, it doesn't need to wait to be cleaved there in the middle because it's already cleaved. And this is mediated because it has this little insertion of these, 4 amino acids there, is mediated by a host enzyme, which is, called furin. There's multiple other enzymes other than furin that will do the same, but furin will do it. So that's why we call it furin cleavage site, because the host enzyme furin can now cleave it right there. And that now means that when, SARS-CoV-2 essentially just needs to bind to a cell, and then once it binds to a cell, it can also infect that cell. SARS-1 can't do that, right? It needs to do these 2 steps. So that's probably the reason why, SARS-CoV-2 can infect many more cells than SARS-1 can, because, it doesn't need to wait to be cleaved. It's already cleaved. It's already good to go. Right? And that's what sort of makes the big difference between the 2. So that was our first sort of thing, saying, okay, what makes this so special, right? And then we wanted to go the next step, which was that, well, okay, but how did it acquire these features, right? And this is why I was pretty skeptical in the beginning, because if you look at the type of research that's being done with these types of viruses, it's, bat viruses, and there's a lot of research to take these bat viruses, they grow them in tissue culture. They do gain-of-function experiments to see what does it take for a bat virus to infect human cells, for example, which typically they can't do in the beginning. And these kind of experiments are done under what's called BSL-2, which is sort of, in terms of just the kind of protection you have is less than, for example, BSL-4, which is why you handle all the really dangerous pathogens, right? And you can do all of this in BSL-2 because it's a bat virus. It's not a human virus until it is, right? But that's so, and this type of work has been going on in Wuhan for many years, like decades, right? It's been going on many other places too, including the United States. But they had specifically at the Wuhan Institute of Virology, they did have a lot of this type of work going on. And that had me sort of worried, because I thought, well, this type of research could quite easily lead to an accident, which would lead to, having evolved the virus in tissue culture, not understanding what was going on, and then accidentally, infecting themselves, and then off the pandemic, right? So I was worried that was going on, and I should say the White House and others were also worried. And there was an established committee, and there was a lot of stuff going on behind the scene on that. But we then started looking at it, because if we say, okay, if we think that these features, how did it acquire that? It's because it's been cultured or created, which we also had to consider, but we found that highly unlikely, right? But we had to consider that part. Or is it just natural evolution, right? So just jump straight out of it. And what is really important here is that by going through sort of this exercise and looking at, these types of features, the first thing was that the fact that this virus binds really well to the human version of the ACE2 receptor, is mediated by what's called the receptor binding domain, which is still in this spike protein. And it's about 5 amino acids which really determines this. But what was seen, and if you look at the bat version of these, receptor binding domains, the RBD here, they're very different, right? They don't really look like what we see in SARS-CoV-2. But then as we were investigating this, people published similar viruses from pangolins, and the pangolins had exactly the same receptor binding domain. It was fully conserved between pangolins and SARS-CoV-2. And what that tells us is that, like the reason why this binds so well to humans, well, that is apparently something that happens in nature too. Specifically, we have seen it in pangolins, so it can happen in all kinds of other animals that we haven't sampled yet. And the other thing was that, and you mentioned this, right, that this receptor binding domain, while it binds really well, there were plenty of papers before that described exactly that if you wanted to design like a receptor binding domain that binds really well to humans, it should look like this, right? And that was described. And this one didn't look like that, right? It was different, but it has found its own optimal way of binding to the human receptor. But again, the pangolin sequences show, this is something that can happen in nature. So clearly, the two unusual features we had discovered there, one of them was already explained as being, this is totally natural. We've seen this before. And that left us with this furin cleavage site, right? So that last feature that made this a human pathogen. And we couldn't fully show that because this is unique to this virus. It hasn't been seen in these types of viruses before, although other coronaviruses do have them. So do flu viruses and HIV and many other viruses have them, right? We said, like, look, we find it's pretty unlikely that would pick something like that up in tissue culture. So we predict that pretty soon, as people start sampling these types of viruses more, we're going to see other viruses, other closely related viruses that have very similar features, either fully formed furin cleavage sites or sort of remnants of furin cleavage sites. And in fact, like I think maybe only a week after we published that paper, that it exactly came out, like a paper showing how in bats, these types of viruses, some of them have insertions in this particular part. So we know that this is an active site, evolutionarily speaking. So things like this come in, and again, in nature. More recently, other research have shown that if you actually culture SARS-CoV-2 in tissue culture, it actually loses this furin cleavage site. So clearly it's not something you would have gained if when you now take the virus that you have, it actually loses that particular site, right? That doesn't make any sense. So all of these things together certainly fully convinced me, and has convinced, I believe, most scientists looking at the data, right, that this is a completely natural virus, right? These features that makes it such an unusual human pathogen, we already see in nature. And once you have sort of made that and convinced yourself that, yeah, actually this is it. And I should say that there was other things that we looked at that were inconsistent with what you would expect in tissue culture. So we could sort of disprove that part too, is that there's no reason to sort of think that there could be a link to a lab here, right? Which the president has been doing, and many other people have been doing too, where they say that, well, but maybe somebody found the virus in a bat and then they put it into a lab, and we know it's not man-made, but then that could have happened. But that can't, I mean, that then it changes from biology to likelihood, right? And the likelihood for that happening versus we get into contact with these kinds of viruses all the time, right? Millions of times every year, there's encounters with these kinds of viruses with people living in China and other places versus a researcher that magically found the next pandemic virus in a bat. But you would hope those types of viruses are really, really rare. If they weren't, we would all be dead from coronaviruses, right? And we're not. So the likelihood of one scenario, the fully natural scenario, versus one where a researcher had picked that virus out of a bat and then accidentally infected themselves in the lab is, I mean, it's a billion to one, right? So that's sort of like all of that together sort of allows us to, number one, this is why it's such an unusual human pathogen. Number two, it's a natural virus.
Mm-hmm.
And that's pretty much the paper.
And I was, really, you mentioned the word skepticism. I think it was your skepticism and very careful science that attracted me to this paper. And, in contrast to some of the very hasty reporting, pangolins, bats, oh my God, you've got an important line in here that says no animal coronavirus has been identified that is sufficiently similar to have served as a direct progenitor of SARS-CoV-2.
Right.
The diversity of coronaviruses in bats and other species is massively undersampled. So refusing to jump to a conclusion, clearly pointing to evidence that these coronaviruses are getting around, but also not, I've looked at the other papers, both in your bibliography and other papers that have come out since. And there are sequencing similarities, but on some of these key binding domains, it just doesn't, like that bat story, that pangolin story doesn't make sense. And one, just like listening to you talk now and having freshly read a paper looking at ACE2 sequence similarities across a whole bunch of different species, basically it found that the pangolin ACE2 is really different from the human ACE2, which leads me to wonder, is that pangolin just infected with a human strain rather than vice, because we like to think of nature as like the source of these zoonotic outbreaks and less about ourselves as like, hotbeds of viral recombination. But that's just a little bit of speculation on my part. There's also a bunch of questions stacking up in the chat, and I don't want to hog it, but maybe just a comments on that point.
So those pangolin viruses are really quite different, right? So what's interesting about these pangolin viruses is that they're only about like 90% or so identical to SARS-CoV-2, except for in the receptor binding domain where they're 100% identical. We can sort of look at that, and we think it's because of recombination probably in a bat species. Coronaviruses recombine all the time, which is a problem because they get a good chance to sort of mix up genetic material, right? And it appears that SARS-CoV-2 is quite clearly that, or some common ancestor of SARS-CoV-2 was a recombinant. We don't think that it went from humans into pangolins. The pangolins got a very similar disease to humans. The paper, that paper is from one of my old colleagues at the Broad, Eleanor Carlson, and I did talk to Eleanor a couple of times about that paper. And what's important is that there are key residues in SARS-CoV-2 and coronaviruses determining binding specifically to the ACE2 receptor. The same is also true in the ACE2 receptor. There are very specific residues that determine specific binding to, the SARS-CoV-2 spike protein. So overall, yes, they aren't that similar. And some of these key residues, they also aren't that similar, right? But again, the way the proteins fold and things like that, that things that you predict, like actually why would a pangolin, ACE2 lead to a receptor binding domain in a virus that would have the same, leading to high human affinity. We don't know why that is. The only thing we can say is that we know that happened, right? And I suspect it has to do again with folding other proteins, and not when you do all these analyses, right? You're just looking at the amino acids and you align them, and you look at that in a 1D structure. But of course, all proteins like this have quaternary structure, right? Which we can't really predict. And I suspect there's something going on there which we just don't understand. I'm pretty sure that there are probably structural labs working on, these different, questions, because you can take the pangolin ACE2 and take the human ACE2, you take SARS, you can take SARS-CoV-2, you can bind them together and you see how optimal is it actually, which I think would be interesting.
So going back a few steps, Michelle, Rachel, and Emma have variations of a question about the spike protein and what makes it infectious. Do any one of you three want to kind of ask that question that's in the chat? And I don't know if you can see the chat question, but it pops up on the side.
Yeah, so thanks, Kristian. It's great to hear the explanation. So I suppose it has got two questions. One is just a question about the definition of infectivity. And so it sounds like from what you were describing, the ability of this furin cleavage, pre-cleavaged, that's what makes it so infectious, that it's so effective at infecting cells in the body. So I just wondered, like clinically, like when they say, you know, SARS, that COVID is so infectious, I suppose people imagine that, you know, it's very good at moving between people, or, you know, there's been a lot of press on how it survives on surfaces. But are you saying that the infectivity, is it primarily from its ability to infect cells within the body, or is it a combination of its survival on surfaces?
Well, so it's everything, right? So if you look at like the stability of SARS-CoV-2 on multiple surfaces, yeah, it's pretty stable. But it's as stable as SARS. So SARS had, you know, almost the same stability. If you look at those papers, you'll see that SARS and SARS-CoV-2, they're essentially identical. Right? There's a few differences, but generally speaking, they're the same, right? Yet SARS doesn't spread very well, right? And the reason for that is that, you know, when we're thinking about droplets, and there's some airborne infections too of this virus, likely, it's because the virus is so high up in the airways, right? So if you just cough, you speak even, right, that's enough to spread the virus. For SARS, that's not enough because it's all the way down into the lungs. So for you to actually spread SARS, you have to really be coughing and sort of like having a lot of stuff coming up, right? In which case it will spread. So if you're exposed, if you take a cup of SARS or a cup of SARS-CoV-2, right, they would be probably be equally well at actually infecting you, right? But again, for me to spread to you is something that can happen really easily, just because it's so high up in the airways. In fact, it's all the way into your mouth, right? And then a major difference also, just in terms of like how these sort of epidemics unfold, right, is that for SARS-CoV-2, you start transmitting the virus several days before you have any sort of symptoms. And that's because like it starts infecting your mouth and your upper airways and whatnot, right? And you probably don't have any symptoms because, you know, that doesn't really lead to any systemic disease. There's no virus in the blood. You probably even haven't noticed that you have the virus, right? But now it sets exactly where it wants to sit in terms of transmitting, right? Because that's where it transmits from. So that's why when we're talking about, you know, SARS and SARS-CoV-2, that's the main difference. Although it also comes down to like when it then enters the body and you have a cell that actually needs to be infected, SARS-CoV-2 is better at doing that because it has that protein cleaved already, right? Versus SARS that doesn't have that protein cleaved. So SARS-CoV-2 can go directly in and infect. It doesn't need anything extra. It always already has what it needs. SARS needs other enzymes too to be active. Otherwise, it can't fuse into the cell and actually cause infection. So it's all these things together that means that SARS-CoV-2 is so effective at spreading and is so infectious because of it.
I've just got two more quick questions, if that's all right, Evan. One is just kind of a follow-up that, so, you know, our saving grace is that there's not very high mortality, well, compared to the flu, but not compared to other pandemics. Is there any evolutionary pressure that would make SARS-CoV-2 more lethal to humans? I can't think of a reason why there would be, but I'm interested in your...
Yeah, no, I mean, there's sort of the, you know, commonly people will say that, you know, viruses will evolve towards being less, you know, less lethal. That is not really true. So there isn't really any strong selection pressure on a virus to say like, no, you shouldn't kill so many people, right? And it's really dependent on what the virus is. For example, Ebola, it's the opposite, right? Because Ebola primarily spreads, early in outbreaks, primarily spreads via funerals, right? So Ebola, the selection pressure is actually there to be more lethal because those are the ones that will get to spread because, again, it primarily spreads via funerals. Right, so it's fully dependent on like what is the pathogen and what is the mode of transmission. For something like SARS-CoV-2, it doesn't care how sick you get. It can spread, probably primarily spreads before you even get sick, right? If you get sick later on, well, tough luck, right? SARS-CoV-2 doesn't care because it's already spread. So there's no selection pressure, I would say, significant selection pressure towards being less lethal. But there's also no selection pressure towards being more lethal, right? It's just a random, like the selection pressures there are, is that there might be some selection pressure for transmission. Something that can transmit better is more likely in a pandemic setting, right, to be more successful than something that cannot. But SARS-CoV-2 is already highly capable of spreading and transmitting, right? So there's probably not really a very, very big selection pressure there. The main selection pressures are how well do you actually survive inside an infected human. And that has to do with the immune system. It has to do with the human, versions of ACE2, things like that. That's where the main selective pressures are. The rest is just drift, right? So I would say for SARS-CoV-2, it goes, either way. I would say, though, that just in terms of lethality and mortality of this is that this is very severe, right? It is, we still don't quite know what this is, but if you just look at, from the sort of the statistics on the number of cases that you count, right? There's going to be way more cases than that, but for the cases that you count, something between like 10 to 15% of those will die, right? If you go and do the calculations now in the the United States, it's probably like 5%, but there's a time delay, right? Because it takes about 5 days to be a case. It then takes another 10 to 15 days before you die, right? So the denominator on whatever deaths you have today, it's what happened 2 weeks ago, right? So when you adjust for this again, the fatality rate on the cases that you count, right, ends up being really high, 10 to 15%. For something like flu, it's much, 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 much lower. I mean, the typical is that people will say that this is maybe 10 times as bad as flu in terms of mortality. It could very likely be between 50 to 100 times as far as I'm concerned. Now, what's really important is that this 10%, 15%, whatever is overinflated, right? Because there's lots of infections that we miss. We don't know exactly how many, right? We don't know exactly, there's also deaths that we miss, right? Certainly. But again, the deaths are, it's more easy to count death and somebody dying from disease than it is counting somebody who just get infected and never knew, right? So the true case fatality rate for this is probably, I would say, in the order of like 3% or so. The infection fatality rate, so these include people that never have any symptoms at all, is probably about 1% or so, which is, I would say, about 50 times higher than flu, right? So in terms of that, like, so personal risk, as long as you're, younger, is very low, right? If you're older, it's very high. If you're older and comorbidities, it's very, very, very high, right? But as a population, risk is enormous, right? Because if you end up killing, in the best case scenario, 1% of your population, but in the worst case scenario, because if you overrun your hospital system, that 1% probably gets closer to 3, if not 4%, is catastrophic, right? So we do need to keep that in mind in terms of when we're thinking about this virus, right? That these look like small numbers, and if you think that flu is 0.1% and SARS-CoV-2 is 1%, people are like, well, but that doesn't seem so different, right? But it is. It's 10 times as bad. That is pretty bad. And then there's no immunity in the population at all, right? So, and the R0 is much, much higher, right? So everybody can get infected. And that makes a huge difference, again, from a population perspective.
Celia, you had a couple of questions if you wanted to ask them.
Thank you, Eben, and thank you, Kristian. First, I wanted to say about your article, I really appreciated that you brought up the issue of dense populations and industrial agriculture when you were talking about the avian. When I was working on this a decade ago and reading everything about it, there was a real cleavage between the critical social scientists and the natural scientists who just dealt with the morphology, but didn't get into any kind of hints of, you know, how the human world is intersecting with the creation of virulent diseases. So I just want to say that. But also, let's see. I wanted, so really briefly, did I hear this right, that this is the first time, when you said this is the first time a non-flu virus has done this, I mean, there's the plague. I wasn't sure what this was that you were referring to. But then here's my main question. So you've been presenting information about morphology and binding to a receptor. But I also have in my mind this new research that's talking about the live polio vaccine, and that is seeming to be talking about some kind of processes that create a more generic immunity. And if those processes exist, are they operating? How do they fit in with this very intimate morphology that you have been describing, where it's like a lock and a key and two things exactly have to fit together? How do you develop this you know, and then I guess another way of asking this question is, what does this all look like if you're a researcher who's working from the perspective of the body and not the virus? Like what key things do we have to think about biologically or morphologically or in any other way when we try and imagine the body mounting a resistance to this lock and key and two-step process that you mentioned? So those are my questions. Thank you.
Yes, I think they're pretty broad. So we covered all of biology right there. So let me take so when you're talking about the vaccine, are you talking about the TB vaccine, not the polio vaccine? That there's been speculation that, you know, pre-vaccination with the TB vaccine might actually protect you against SARS-CoV-2.
Yes.
That's the one. Yeah. Okay. Yeah. So there's certainly been speculations around that, and I don't think they will turn out to be true. I think it's just an early correlation that people noticed because certain countries were delayed in terms of starting their own epidemics, right? So like you look at Russia, for example, it's like, oh, it doesn't seem like Russia, you know, gets this virus, right? But they do, and they have it now. And we'll see it explosive spread in Russia too, like we will everywhere. So I highly doubt that, you know, the pre-vaccination with you know, the TB vaccine will help protect you against SARS-CoV-2. In terms of, you know, natural immunity, you know, there's other coronaviruses where there might be some cross protection, right? There is the common cold coronaviruses that we all get hit with every year. And one big question is that could there be some cross protection in a subset of people because of that? And I mean, my best guess is that probably not, but you know, there is that possibility. In terms of the virus, the body adapting to the virus, there is no adaptation, right? There's immunity. So if you get hit by the virus, we hope that you develop immunity to the virus, but we don't actually know if that's the case. And even if we do, you don't know for how long that'll last. And these are, you know, big problems because there's a lot of people talking about herd immunity, you know, natural induced herd immunity, where you sort of slowly let this virus, you know, move through a population and the population over time will become immune. But we don't actually know if that works. We don't know if that, well, we know that as a strategy itself does not work, but for many different reasons. But we don't actually know if immunity like that is lasting. And the reason why I say that is that if you look at the common cold coronaviruses, our immunity to those only seem to last a few months. If we look to SARS and MERS, which are more like SARS-CoV-2, it seems to maybe last a couple of years, which is pretty short. Right, a lot of different viruses you get infected with, you have much longer lasting immunity. But of course, there's all the other viruses and pathogens like malaria, for example. You get hit by that every year and you never have immunity, right? You get hit by hepatitis C, for example. There's no immunity, it just stays with you, right? So there are many examples of both viruses and other pathogens where, again, immunity is fleeting. So my best guess is that people that are infected will have some level of immunity, but I don't know for how long. And I also don't know if that works in people that are fully asymptomatic, for example. I think a big question we need to know is that if you're fully asymptomatic, do you actually have immunity to this virus? And I'm worried that the answer to that is going to be no, right? But we don't know yet. That's too early, right? In terms of sort of the body adapting or the virus, if we just give it time, the virus won't be as bad, or the human body will be able to deal with it. That's not going to happen, right? Like the virus is not going to be any less lethal than it is now. And we as a species also won't be able to better tolerate this, give it time. And because there are people thinking that, our president is saying it'll probably just go away, right? And it won't, right? We tend to sort of, because we draw to the flu scenario, right, where flus come in and then big epidemics and then it goes away, right? And we sort of think of this as maybe it's the same, but the reason for that is that we have lots of pre-existing immunity to flu. We have vaccines, a lot of people get vaccinated, right? And that sort of makes it go away, right? And it's a much more mild disease. This, again, is completely new. There's no pre-existing immunity whatsoever. If we look at the immunity levels in most population right now, it's very, very low single digits, right? Probably 2 to 3%. So a place like San Diego here, probably around 1%. And that's low. That leaves a big gap of people that can still be infected.
So there's a lot of questions stacking up about, oh, go ahead, Susan.
I'll just say thank you. Yeah, my question was really about immunity, not adaptation.
Yeah,
so,
yeah.
But you answered it. Thank you.
Astrid's been trying to get a question in for a while. Yeah.
Great. Go ahead, Astrid.
Hi, thank you. Thanks a lot for all these detailed explanation. I would like to come back to something you mentioned at the very beginning, and I'm not quite sure I followed all the details. I'm very interested in the investment of this virus being natural, and you explained why it is unlikely that it didn't escape a laboratory. I have been wondering when the virus jumped, at the moment it jumped species, that it mutates somehow within a human host, right? How do you know what are the properties of the virus and what is acquired within some kind of rearrangement that happened when it mutates within a host which is a new species for the virus? So how do you know? I'm trying to think of a way to problematize what naturally belongs to the virus or what is acquired in the moment is jumped the species, and how do you know? How do you distinguish that? Which of these property that makes it so successful has been acquired at which stage? If that makes any sense, I don't know.
No, it makes sense. It makes sense, and I think like. In our paper, we entertain two different scenarios, right? We entertain the scenario in which the virus jumps directly from an animal host. That can be a bat, it can be an intermediate species, probably an intermediate species because bats are, hibernating in the winter months in Wuhan and other places, so that seems unlikely. But SARS and MERS and others, they come out of intermediate hosts, right? So one we entertain is that, the virus evolved in this host and then it found a human and jumped directly. There's no mutation, there's nothing, right? As soon as it starts spreading in humans, very early on, there's a couple of fixed mutations that happens because they're selected for, but they're minor, right? So basically the virus jumps fully formed directly from an intermediate host into a human host. We know based on the genetic data, because we can, analyze all of this and say, when did all these viruses we see circulating now, when did they have a common ancestor? This is based on molecular clocks and things like that. And we can show that that's like mid-November to early December, right? So right around the time that we know from caseworks, epidemiological investigations, right? And we know that the outbreak started then. So all of this is fully consistent. The other scenario that we entertain, which I would say is probably a little less likely, but is still a plausible scenario, is that a virus, for example, without this furin cleavage site, jumped into a human, and then the sort of low-level spread in humans, which, if it doesn't have that furin cleavage site, probably wouldn't really lead to any disease, right? So people would probably have mild disease or no disease at all, and it would never have been detected, right? Because there's a lot of people, and there's only so many ways you can survey them. But then at some point in some individual, you can imagine maybe that has like, remnants of that furin cleavage site for other reasons, right? Like evolution will just give you random stuff, right? At some point, maybe that furin cleavage site evolved in a single host, and then from there fully took off, right? And then that dating event we can show again, which is mid-November, mid-December, becomes the acquisition of that furin cleavage site. So, we can't say with any sort of certainty, it's definitely this, it's definitely that. This is when it picked that up, this is when it picked that up, because that will require us having historical records of this virus moving from a bat to an intermediate host and into humans. And you can never do that, right? Because there's so many bats and so many intermediate hosts and so many different viruses out there. And you can't hope to survey even just a fraction, fleeting small fraction of them, right? So that can be done. Now for SARS was a little different, because SARS, we basically discovered exactly the same viruses in civets and raccoon dogs, and SARS was more widely distributed in an intermediate host. So for there you can go and say, oh, there it is, right? So that fully proves it. For SARS-CoV-2, because we don't have that captured, sequence from intermediate hosts, we can't make the same conclusion. But that's not weird. It's just because, again, there's so many different hosts that it could have been in. And again, we can only sample a really, really small fraction of it. And it's very likely that if that intermediate host actually got sick with the disease, like pangolins do, for example, they might also have died off. So there's currently potentially no intermediate host where you would actually find the virus, right? And again, that's not mysterious. It's not weird. It's just a limitation of science.
May I ask a follow-up question to the second scenario? Could that have happened with a virus that escaped from a laboratory?
I mean, again, you can't, prove a negative, right? So, and this is the problem we keep running into, because people saying like, well, but it could have been a pre-virus that escaped from a laboratory. It could be a fully formed SARS-CoV-2 that escaped from a laboratory. What I can say is that the likelihood of that, compared again to the natural scenario where people get into contact with these viruses all the time, is incredibly small. What we do know is that for all the public records, for everything that the Wuhan Institute of Virology have ever sequenced, and they've sequenced a lot of these viruses, this one is very distant to any of those. Even the bat virus, that's 96%. That is like 20, 30 years in evolutionary time back in time, that there's no way that could have served as the precursor for this virus, right? So when you sort of, looking at these two scenarios, was it, does it come out of an animal, or did it come out of a lab? We have tons of data showing that it very likely came out of an animal. We have zero data suggesting it came out of a lab, right? There's only speculation, and that speculation is like, there's a world-class virology institute there, and it happens to be within 20 miles of a wet market that's there, right? That's not data. That's a coincidence, right? So it's really important, again, when we think about these things, that, you can't disprove a negative. So I can't, 100% prove that, no, it definitely didn't come out of the lab. I can show you there's no data to support that. There's certain versions of the lab scenario, like the culture, for example, which I can disprove based on the data, right? I can disprove that it's not a bioweapon, it's not an engineered virus. I can disprove these things, right? But I can't fully disprove that a slightly different precursor virus or this exact virus came out of the freezer in a lab. All I can say is that it's hugely unlikely, right? Again, this is why I'm saying like one is like, it's 1 billion to 1, right? And that's really important to keep in mind.
So Zoe has a question. Do you want to ask it? Does COVID interact with other viruses?
No, it doesn't interact with other viruses, but it does, like these things do recombine, right? So if you find SARS-CoV-2 in, you know, a cell with another version of SARS-CoV-2, there's only one version, but let's imagine there's a little, because there's genetic diversity in the population of these, right? So they're all slightly different just from their genetics, not from their function. But, you know, it is possible that you will see like some of these happening where they will exchange genetic material via recombination. And it's just because SARS coronaviruses in general just recombine a lot, right? So things like that could happen. It wouldn't really result in a different virus because they're all, you know, pretty much identical. But, you know, there are no like other circulating SARS-like coronaviruses, right? That it could find itself in that host. But if you go back to like the bat, you know, the bat species where these guys live, they exchange genetic material all the time, right? They recombine all the time. And this is how we think we ended up with this SARS-CoV-2 as well, right? But not in humans. And there's no like interaction with like flu and HIV and other viruses because that's not compatible with biology.
So when you say there's no recombination events in humans, I mean, in my mind, you know, like if there's 2 or 3 viruses in a given cell,
yeah,
it doesn't matter if it's a bat cell or a human cell, like the same recombination events could happen.
Yeah, no, for sure. That could happen, but like that, you know, virus number 1, 2, and 3, it couldn't be 3, but it could be 2. Mm-hmm. Right? I think that's rare, but it does happen given how many infections we have. Those 2 versions of the virus would essentially be identical. Mm-hmm. Right? So because of that, there might be a recombination, but if you have, you know, an almost identical virus, and when I say almost identical, I mean, they might differ by like 2 nucleotide differences in like a 30 kb genome, right? That if they recombine, you essentially end up with exactly the same, right? So it doesn't lead to like, but that is different in like in a bat, for example, where you can imagine that you have SARS-CoV-2 and SARS-1, right? They're 80% identical, so they're quite different. They could find themselves in the same cell, and now they can recombine, and what comes out of that is truly a different virus, right? So that's the scenario that happens all the time because these bats and other animals too, right, are chock full of SARS-like coronaviruses.
Industrial pigs, for example.
Yes.
We haven't really talked about the other article, which, you know, is finding the earlier SARS in you know, pigs a few years after the original SARS outbreak.
Yeah, I mean, there's the idea that because there's industrial farming, right, of animals in China that do carry SARS-like coronaviruses, right? And I think there's a lot of people that think that could have been one of the reasons why we've started seeing the emergence of this, maybe even of SARS itself, right? But definitely a lot of hypotheses around that with SARS-CoV-2. Mm-hmm. And I find that as a plausible explanation for potential intermediate hosts here, right? Which is pretty terrifying because as we do more of this, deforestation, population growth, all these kinds of things, we might expect more events like the one we are observing now to, you know, increase in frequency in the future. I should say that, you know, it's important to remember that like this has been the case for flu viruses all along, right? You go to hog farms in Iowa, or whatever you go. I mean, I'm from Denmark. We have 10 times as many pigs as we do humans in that country, right? That they're in there, viruses recombine and reassort all the time. You have the bird flus coming in, and you have the avian flus coming in. You have human flus coming in. All sort of serve as a mixing vessel in pigs, right? And that's how the 2009 H1N1 pandemic, right? Those were Mexican pigs, right? So these things can happen. It's just it's a different kind of virus, of course.
Rachel, you had an allied question, then Bettina.
There's another stack as well.
Oh, there's another stack. Okay.
Yeah. I think the order would be Roberta, Anna, Emma, if you want to, and then Rachel and Bettina.
So, Kristian, thank you very much. It's really interesting. I'm from Italy, actually, and I live a couple of hours from Lombardy, the region that has been most affected here in Italy. And so there are different hypotheses, and scientists are trying to understand why this happened so specifically in that region. And I would like just to know if you have any explanation, any hypothesis.
Yeah, I mean, I've gotten that question often, and all I can say is that I think, you know, it's a combination of just a, you know, random chance, but then also there must have been a lot of traffic to, you know, that region, maybe via Milan, right? That's a big city, and from there, you know, spread further north and other places, right? Why was it Italy? I mean, it had to be a different place, right? Italy did not look for the virus. So by the time Italy, like America, right, same situation, by the time that they realized, you know, we have SARS-CoV-2, it had been there for a while, right? And because of exponential growth, that if you don't realize this early enough, it will look like it'll explode really fast because it's, you know, doubling every 2 to 3 days, right? The other thing is that Italy, you know, you have a lot of multi-generational households, which might, you know, be an easy way for the virus to spread because there's a lot of old people living with young people going out, doing the business, getting infected, not really realizing they're sick, and then coming back and then infecting other people in the household. There are other countries like Spain, for example, Greece, certainly even more so, right? Where that is true, where it didn't take off, but it then later did, right? So I just think that Italy was unlucky of sort of being where the virus found itself first in an unprepared country, like all other countries in Europe and certainly the United States. And that's why it took off, right? I don't think there's anything special. There's just a set of factors which also exist in other places, and then just a random luck of the draw, right? Saying that that's just how it ended up.
Yeah, okay, thanks.
So it's okay, we'll skip you, Emma, because you already got to ask some questions. Go to Rachel and then Bettina. Can't hear.
Rachel, you're on, I don't know if you're on mute.
We'll go to Bettina and then Rachel, if you can figure out how to unmute yourself.
Yeah, maybe try and take off the headphones. That's sometimes work, at least for me. I think Bettina is, are you?
I'm unmuted now. Should I?
Yeah, go for it.
Okay, yeah. I had two questions. One is actually related to a question that Emma had earlier about the Wuhan lab. And I'm just wondering whether you have any maybe references, you know, because we don't have that much time to kind of understand the history of the Wuhan lab a little bit better, because Emma had a question about, you know, that it just seems like such a huge coincidence, you know, that the virus kind of
but I don't know. Let me take the coincidence, right? Because what we're observing is such a rare event that as humans, we're trying to look for explanations, right? We already have the explanation, which is a jump from an animal into humans. Right. But there's this additional need that's saying like, yes, but it seems like, you know, strong coincidence, there's a lab there. And I don't really know if that is the case. Like, to me, that's not very coincidental. If you look at SARS, that happened pretty close to Wuhan, right? That was a province further down, and was also a wet market. So, you know, I don't think from that perspective, it's like, yeah, there is certainly coincidental that you have this world-class, you know, biology institute that do happen to study, you know, bat viruses. But the reason why they're studying bat viruses there, these SARS-like coronaviruses there, is because they have a lot of SARS-like coronaviruses there, right? And because they have a lot of SARS-like coronaviruses around there, that means that the likelihood of something like this happening is just much higher.
Okay.
Right? So I would say that if the Wuhan Institute of Virology was in a place where there were no SARS-like coronaviruses whatsoever, imagine it was in San Diego here, and then all of a sudden you have a huge outbreak of, you know, SARS-like coronavirus where there is a lab and you don't have SARS-like coronaviruses around, sure. that would be, you know, raise an additional red flag here. Right? But again, the coincidences here, which have been talked about a lot, right, are just that. They're just coincidences. I would say from my own perspective, I'm really happy that, you know, this happened in Wuhan, because the reason why we learned so much about this virus so quickly, including the identification of this being a novel coronavirus, they only knew that because they had all the experts there that knew what this kind of disease looked like. They had the diagnostics in place to, you know, because we don't have any specific diagnostics for this virus before we know about it, right? They had those in place so they could do the identification really rapidly. And then only within like weeks, we had the genetic sequence, so we can start developing specific diagnostics as well as vaccines and start all of that work, right? As well as like understanding what is the receptor, so we can start developing drugs and take the drugs out that we know works against these different interactions, right? And a lot of this was accelerated because Wuhan Institute of Virology is there. Right, so I would say, like, when we look at it like that, I'm really happy it happened there, not somewhere else, where you wouldn't have a bunch of experts around to immediately jump in.
Yeah, yeah. Can I ask my second question really quick?
Real quick.
I think it's going to not require much, but since you mentioned Denmark and the pork industry, you know, I've been recently following the spread of African swine fever, and Denmark built a huge border fence.
Build a wall.
What's that? A wall to keep the wild boar out, which are considered to be vectors to transmit the disease. And so I'm wondering whether, because you just were talking about that, whether you see a danger, for example, for diseases like African swine fever to also become zoonotic diseases.
I mean, you might have limited sort of transmission, right? But generally speaking, luckily, right, is that most viruses that live in other animals don't jump into humans all of a sudden. And it's because viruses are really rather dumb, right? They most of what they need is in the host that they're trying to infect. They don't have it themselves, right? They rely entirely on the host machinery to replicate. And the versions of whatever you have is just so different, right, than what you would find in a pig or what you will find in a bat or whatever. So it just doesn't work very efficiently. So while you might have and this is what happens with the bird flus, for example, right, they don't really like the human host very well, right, because it doesn't fit very well. So, you know, for most of these other ones, I would say it's the same. Right. That's not to say it could never happen, because anything can happen, right? But I think that's pretty unlikely.
Rachel, do you want to try again?
Can you hear me?
Yeah, we can.
Okay. Thank you. I was just struck by, as I was reading your piece and then the other piece that was I think it was from about 2004 that we prepped for today. And then just as somebody who's from a very rural, economically impoverished place in the Midwest, that was struck by the epidemic diarrhea virus among pigs. Just thinking, I guess, about the careful way in which your piece was looking at kind of a viral memory of sorts, and the ways in which the agricultural industry, as I've been reading around this, just even over the last few weeks, is really attempting to kind of say, well, we're not seeing this in pigs, or we're not seeing this in poultry. And on the one hand, you might think, well, I guess that's just being extra careful. But on the other hand, I guess I just, in correlation to reading your piece and then reading the other piece that we read from a mere however many years ago, and then thinking together with the 2014 outbreaks, it seems like very bad practice as well. I can't, on the one hand you think it's careful, on the other hand you think it's quite manipulative and quite dangerous on an industrial level.
I mean, I think it is, right? I mean, like flu viruses in pigs, that's a bad combination. And I mean, again, Denmark has a flu surveillance program in their pigs, right, where they sequence the viruses out of pigs. And I mean, they see crazy stuff, right? I mean, there is recombinations and reassortments like you have never seen. It's just most of these viruses are non-functional in terms of being able to transmit to humans. But then every once in a while, you just have that lucky combination of, segments that actually can transmit to humans. And then it starts a pandemic. That's exactly what happened in 2009, right? But in Mexico, but it could be in Danish pigs too the next time, right? So, we know these things happen. We haven't really seen like a lot of evidence for SARS-like coronaviruses in pigs. So I would say that that's not my main concern. Wouldn't be pigs specifically, but there's a lot of other industrial farming, right? I don't think civets, but I think like some of the raccoons and ferrets and things like that, there are big industries around that in terms of farming, right? And that could certainly present, a risk for things like SARS-like coronaviruses, but very similar to what flu might do in hog farms. And, it can be anywhere. Again, it happened to be in Mexico. Typically, seasonal flu viruses come out of Asia, right? But these pandemics can come out of anywhere and any time. Right? They happen with some occurrence, right? But yeah, the more we have this kind of industrial farming and really close connection, also deforestation and people moving into the, further into the jungle, if you want. I mean, that's probably what led to like the 2014 Ebola epidemic in West Africa, right? Because Ebola had probably been there for quite some time, but people just hadn't gotten into contact with it. And as you move further into the forest, or you have bigger farms, mill farming around, animal farms where human viruses can live, then yes, the risk is definitely increased. No question about it. All right, so I actually have to get on.
We could bounce too, but we're very grateful for you joining us.
This was fun.
I think this is an example of like excellent science that we've seen a lot of science that's kind of parroting kind of cultural stereotypes and things like this, and I think what stood out about yours is just the very serious, careful, meticulous work that you were doing. That I just can't say enough how much the article was important for this conversation we've been having. So,
thanks.
So thank you.
Yeah, and thanks for reading it.
Yeah. All right.
Good talking to you guys.
All right.
See you later.
Ciao. Take care. Thanks, everybody. And next week we're switching to Sunday with Keck. Keck is giving us his piece about avian sentinels. So 9 a.m. It'll be better for people in Europe. I think it's 3 o'clock Paris time, 9 a.m. US time. Probably virtually impossible for people in California or Seattle, 6 a.m. unless you get up really early. But Elizabeth will send out the details probably on Monday. And yes, we will record it so people on the West Coast can get the instant replay. All right, thanks everybody.